Assalamualaikum. Today we are going to present about experiment 12, gravimetric analysis of phosphorus in plant food. This is our team members, which are Khairina and Jaznadia. The objective of this experiment is to determine the content percentage of phosphorus in plant food through gravimetric analysis.
First, the plant food sample was obtained and the set of numbers written on the label was recorded. The plant food sample was weighed between 3.0 to 3.5 gram using a weighing paper, and the sample was transferred to a 250 ml beaker. A volume of 35 to 40 ml of distilled water was added, and the mixture was stirred with a glass rod to dissolve the sample. If the sample did not completely dissolve, the insoluble material was removed through filtration. For filtration, about 40 ml of 10% magnesium sulfate heptahydrate was added. Then, about 100 ml of 2 molar ammonia was added, slowly while stirring. A white precipitate of magnesium ammonium sulfate hexahydrate was formed. The mixture was allowed to sit at room temperature for 10 minutes to complete the precipitation.
A filter paper was weighed accurately. The paper was folded and fitted into a glass funnel. The paper was wetted with distilled water to hold it in place in the funnel. The precipitate and all the solution from the beaker were carefully transferred onto the filter paper. The precipitate was washed by adding 2 to 3 portions of 5 ml distilled water to the beaker. The precipitate was then rinsed again with 2 ml portions of 75% isopropyl alcohol through the filter paper. The filter paper was removed, placed on a numbered porcelain evaporating dish, and stored to dry for 2 to 3 days on the bench or dried in the oven for 24 hours at 60 degrees Celsius. When the magnesium ammonium phosphate hexahydrate was thoroughly dry, the filter paper together with the magnesium ammonium phosphate hexahydrate was weighed. The mass was recorded and the percentage of phosphorus in the original sample was calculated.
The plant food name that was used in this experiment is from Bajaj Special Green, with percentage of phosphorus content being 15% and mass of sample used was 3.3328 gram. Below shows the result that was obtained from the experiment, with mass of magnesium ammonium phosphate used being 1.2760 gram. Using gravimetric factor and mass of magnesium ammonium phosphate, we were able to calculate the mole of phosphorus and phosphorus pentoxide. Gravimetric factor of phosphorus can be identified with dividing the molar mass of phosphorus with molar mass of magnesium ammonium phosphate and multiplied it with the mass used based on the data recorded. And finally, mole of phosphorus can be identified. Percentage weight over weight of phosphorus pentoxide can be found by dividing mass of analyte with the mass of sample and multiplied it with 100%. And from that, we had obtained 19.79% of phosphorus pentoxide. Percentage error can be calculated with initial percentage written on the label minus with our percentage yield and dividing it with initial percentage and multiplied it with 100%. From this experiment, we had obtained over 31.93% of error. The phosphorus content of Bajaj Special Green was determined using gravimetric analysis. The experimental result obtained was 19.79% with weight over weight, which is higher than the value stated on the product label, 50% weight over weight. The calculated percentage error was 31.93%, showing a noticeable difference between the two values. This difference may be due to experimental errors such as incomplete washing or drying of the precipitate, which could increase its mass. Errors during filtration, transfer, or weighing of the precipitate may also have affected the accuracy of the result.
This experiment was conducted to determine the percentage of phosphorus in plant food through gravimetric analysis. Gravimetric analysis is a very accurate method to analyze percentage of analyte from a sample, as it does not require standardization and calibration, but mainly focuses on accurate analyze weighing. The sample was in a solid form initially, then dissolved to a liquid, filtered to the solid part, and finally dried to obtain the final precipitate of the analyte. This method is less messy and can be done with a macro sample.